A very good evening. I'm Dr. Karma. The topic for my presentation is antibiotic resistant Acinetobacter baumannii in a tertiary care hospital setting in Sikkim, India. Problems and alternative approach with special focus on natural products. So, as we know, Acinetobacter baumannii is an inescapable pathogen that is capable of causing both community and healthcare associated infection. Now, this organism has recently emerged as a major cause because of its tendency to cause resistance and has a proliferity for pan-drug resistance and can cause large outbreaks in many institutes. Acinetobacter baumannii, it also causes many infections of the cerebrospinal fluid, lungs, wound, urinary tract, skin and soft tissues, and bloodstream, with high rates of morbidity and mortality. A. baumannii has topped the WHO list of 12 bacteria for which new drugs are urgently required. What we see is the irrational use of antibiotics and selective pressure that has led to the Acinetobacter emerging as an antibiotic-resistant pathogen both in the hospital as well as in the environmental setting. Now, mm, these pathogens they are found to persist over a long period of time on inanimate surfaces, especially under a wide range of environmental conditions. Now, this is mostly attributed to the fact because they are strong biofilm producers. And however, what we see is the scant information is available in regard to this, to the chronorelatedness and to the diversity of the A. baumannii strains on the Indian subcontinent. Now, the genomic analysis of India MDR A. baumannii has indicated the presence of interferons, transposomes, and other mobile genetic elements such as Acinetobacter baumannii antibiotic resistance island. So, mm, furthermore, what we see is that this organism they have many or they are known to possess virulence factors that helps in quorum sensing and regulations, which is like the adherence, the biofilm production, serum resistance, and the drug efflux. So these factors, this virulence helps the pathogen to colonize the host, evade or suppress the host immune response, and so. So recent surveillance has indicated that A. baumannii causes around less than 2%, around 2% of the healthcare associated infections in United States, as reported by Bullets et al. In 2018. But the prevalence, what we see here in the southern and the Southeast Asia, is much higher, and they are mostly associated with nosocomial infection, as reported from a study by Suvantarat and Carroll in 2016. Now, carbapenem, they are considered to be one of the drug of choice for treating Acinetobacter infection. However, mm, the increased resistance to this class of antibiotic have been reported worldwide. And what we see is also India reporting about 40 to 75% of resistance rate of Acinetobacter baumannii. So the successful development of efficient antibacterial require a better knowledge of the resistance mechanism, thereby to increase the chance to discover new chemical structures with ideally new mechanism of action or different antimicrobial combination. Now, as a result, WHO has identified carbapenem-resistant A. baumannii or CRAB as the greatest bacterial threat to the human global human health, and it is also considered and has placed it as a top priority for the priority pathogen for the development of new antibiotic. Now, the high prevalence of those strains, these kind of strains, they are resistant to almost all the antibiotics, especially those well tolerated cephalosporins and the carbapenems. Okay, the resistance has emerged. So now this has led us to the revival of drugs considered to be the last resort, like the polymyxins for systemic administration. However, what we see is the resistance to cholesterol is also now becoming more prevalent, and polymyxins are now used less widely due to their serious side effects that are associated with these agents. There are the mechanisms that are associated with the airborne resistance, like beta lactamase expressions, the cell membrane permeability alteration, if there is the efflux pump, if there is an increased expressions or the DNA guidance mutations that we see, and the topoisomerase encoding genes. Now, the most frequent causes of this enzymatic resistance in A. baumannii are represented by the acquired carbapenem hydrolyzing class D beta lactamase like the OXA 23, 24, 58, and so on. Also, the overexpressions of the intrinsic OXA 51. Okay, and these are also there are also another group of class, that is the class A beta lactamase like TEM, SHV, CTXM, KPC, mm, and we also have the metallo-beta-lactamase, the MBL, like your IMP, VIM, SPM, and the NDM, New Delhi meta-beta-lactamase. So, carbapenems, their resistance in A. baumannii is now mainly due to the class D and class B carbapenems that belongs to the Ambler class of beta-lactamase. Although uh, various mechanisms contribute to carbapenem resistance, the majority is basically due to class D carbapenems like OXA, BLA, OXA 23, 24, 58, and so on. And BLA OXA 23, they are responsible mostly for causing outbreaks and have been reported from various parts of the world. Um, and class D carbapenems in transposomes, they have the ability to spread rapidly in successful clonal lineages of A. baumannii. Thus, the successful development of efficient antibacterial requires what we need a better knowledge of the resistance mechanism. So the study was aimed to investigate the antibiotic susceptibility pattern, the biofilm production of these isolates, perform the RT-PCR for the confirmatory identification and detection of the drug-resistant gene, and also we aim to evaluate the selected natural products against the drug-resistant A. baumannii. Now, before that, I would like to speak brief uh, about my institute. Sikkim Manipal Institute of Medical Sciences is a constituent of the Sikkim Manipal University. We have a mother institute down in KMC Mangalore. We offer medical and allied health sciences courses. We have more than 288 faculties offering 150 medical MBBS students. Our hospital, that is the Central Referral Hospital, is a 500-bedded tertiary care hospital, and we have around 21 specialties and super specialties. Coming back to the study is a descriptive type hospital based study. Location is SMIMS and CRH. Lab work, the samples that we received, all the clinical samples that we received for bacterial culture in the Department of Microbiology CRH were processed in Mekongki agar and high chrome Acinetobacter based medium for the isolation of A. baumannii. So what we see is the uh, Total clinical specimen that we received over a period of 1 year was 18,400, out of which A. baumannii was mostly 44, around 45% of A. baumannii was isolated from the endotracheal specimen, around 19% was isolated from the sputum. Coming to the antibiotic susceptibility pattern, what we see is a wide range of resistance pattern in almost all the group of drugs except for your tigecycline and colistin, where tigecycline exhibited only 2% of resistance, around 3% of resistance, and colistin around 8%. Coming to the biofilm production, the biofilm uh, was performed, biofilm identification was performed by the microtiter plate method. Okay, where we use trypticase soy broth with 0.25% glucose at 37 degrees Celsius. The optical density at 620 nanometer was then determined using the microtiter plate reader, and each assay was then performed in triplicate, and the average optical density was considered. So here we see that the highest biofilm producers were weak biofilm producers ranging around 54%. Non-biofilm producers were only 33%. Strong biofilm producers, however, was only 2%. Okay, then we went forward with the real-time PCR for the identification of the abomini, where we use BLA OXA 51 gene. The primer and the probe details as given. What we found was around out of 319 isolates of Acinetobacter species, 307 were confirmed for Acinetobacter baumannii, whereas only 12 other remaining 12 were other Acinetobacter species. Then carbapenem resistant gene, drug resistant gene was detected from the selected 100 samples of 100 Acinetobacter isolates, where we identify genes like BLA-OXA23, BLA-OXA24, 58 under group class B metallo-beta-lactamase. We went for BLA-IMP, VIM, and insertion sequence ISA-BA1 was also used. Here we see that 96% of the isolate I were detected with BLA-OXA23 like, and 76% was detected with ISA-BA1. The virulent genes, we also use the virulence genes like biofilm associated proteins, BAP gene, which showed only out of 100 samples, 31% were, 32% were positive. For OMP, 91% were positive. Then, in the combination of genes, what we saw was 64% of the gene, both BLA OXA23 and ISA BA1 were found together in on the 64 isolate. Uh, then